So welcome back to Biomindscape. Today we are going to know about the rolling circle method. In this, first we will know about various type of mode of replication like conservative, dispersive and semi-conservative. So conservative replication, the conservative replication where the entire double-stranded DNA molecule it serves as a template for the whole new molecule of DNA, and the original DNA molecule it is fully conserved during the replication. Whereas the dispersive replication, both the nucleotides strand break down, disperse into fragments. They serve as a template for making a new DNA fragment, for synthesizing of the new DNA fragment, and then somehow resembles into two complete DNA molecule. So in this module, each result DNA molecule is interspersed with fragment of the old one and the new DNA. So none of the original molecule is conserved. That's why it is dispersive replication. Then semi-conservative replication, it is intermediate between the two model. Two nucleotides stand, they unwind and each serve as a template for the new DNA molecule. So these are the three proposed model of replication they are conservative replication, dispersive replication and semi-conservative replication. So what are the modes of replication? They are like theta replication, rolling circle replication and linear eukaryotic replication. So individual unit of replication we call it as replicons and they consist of their own origin. So we call this as replication origin. So replication it start at the place that is known as the origin and it continue until the entire replicon it has been replicated. Bacterial chromosome have a single replication origin, whereas if we see a eukaryotic chromosome, it have many origin of replication. So first what happened? The unwinding of the double helix generates a loop. We call it as replication bubble. So it is termed as replication bubble. So the point of unwinding, where the unwinding takes place, where the two single nucleotide strands separate from the double-stranded DNA helix, we call that region as an replication fork. So if there are two replication fork, one of each of the replication bubble, the fork it proceed outwards in both the direction and that's why we call it as the bidirectional replication. So what is a theta replication? It is a type of replication which is found commonly in the E. coli and other organism which process the circular DNA. So what are the steps of theta replication? Let's know. First what happened? The double-stranded DNA unwind at the replication origin. Then it proceeds single-stranded template for the synthesis of the new DNA and the replication, the bubble is formed which have a replication fork at each end. So the fork it proceed around a circle and eventually the two circular DNA molecule are produced. So the product of the theta replication are two circular DNA molecule. The second one is the rolling circle replication. What happens here? In the rolling circle replication it takes place in some virus and F factor of E. coli. So replication is initiated by breaking one of the nucleotide strand. First what happens, a nucleotide strand is break and then the DNA synthesized at that place at the 3' end of the broken strand. At the 3' end of the broken strand the DNA synthesis takes place and the inner strand it is used as a template. So the 5' end of the broken strand it is displaced and there is a cleavage released at a single stranded linear DNA and a double stranded circular DNA. This cycle may be repeated. There is a cleavage forming and the linear DNA may circularize and serve as a template for synthesis of a complementary strand. So what happens? The conclusion what we get is the product of the rolling circular replication are multiple circular DNA molecules which we get in this particular place of some virus and F factor of E. coli. Then there is a linear DNA replication taking place in eukaryotic chromosome. First what happened? Each chromosome contains numerous origin, like origin 1, origin 2 and origin 3, and at each origin the DNA is unwinding, producing a replication bubble. So where what happens, the DNA synthesizing takes place on both the strand at each end of the bubble, and the replication forks proceed outwards. So eventually the fork of an adjacent bubble run into each other and segment of the DNA are fused, and they produce the two identical linear DNA molecule. So the product of the eukaryotic DNA replication are two linear DNA molecule. So what are the difference between the replication model like theta, rolling circle and linear eukaryotic? The DNA template are circular in theta and rolling, whereas if we see the linear eukaryotic replication model, the DNA template is linear. And the breakage of the nucleotide strand, there is no breakage in theta and linear eukaryotic, whereas we can see the breakage in rolling circle replication model. So number of replicons are 1 in theta and rolling, whereas there are many number of replicons in the linear eukaryotic, and in it is unidirectional. Which one is unidirectional? The rolling circle is unidirectional, whereas the theta and linear eukaryotic, they are bidirectional. Even the theta, it can be unidirectional or bidirectional. And the product of the theta replication model is that the two circular molecule are present. In the product we get a two circular molecule, whereas in rolling circle we get one circular molecule and one linear molecule that may be circularized, and two linear molecules are formed in the linear eukaryotes. Thanks for watching. Do like, share and subscribe.